Good morning, students. Today's topic of discussion is competition number 3.1 polysaccharides. Here in this polysaccharides we are discussing mainly about two polysaccharides, that is glycogen, which is a storage form of polysaccharide in animal, and glycosaminoglycans, which forms various structures in our body. Polysaccharides, all of us know that these are the carbohydrates which on hydrolysis gives 10 or more monosaccharides. There are two types of polysaccharides, homopolysaccharides and heteropolysaccharides. Homopolysaccharides are the carbohydrates which on hydrolysis gives 10 or more same type of monosaccharides. Example of homopolysaccharides are starch, glycogen, cellulose, dextran, inulin, etc. Out of these, starch, glycogen, cellulose and dextran are polymer of glucose, whereas inulin is a polymer of fructose. Heteropolysaccharide means these are the carbohydrates which on hydrolysis gives 10 or more different types of monosaccharides. Example includes agar and glycosaminoglycans that we have to study today. The examples of these glycosaminoglycans are hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate and keratin sulfate. Now first we will study the glycogen, which is a homopolysaccharide. It is a storage form of carbohydrate in animal tissue. It is mainly stored in liver and muscles. There is limitation for storage of glycogen in our body. Roughly about 5% of the liver weight and 1% of the skeletal muscle weight of the body is the limit for storage of glycogen. Glycogen is a monomer of alpha D glucose. It is a branch structure and having high molecular weight. This is the diagrammatic representation of glycogen molecule, showing each dot as alpha D glucose molecule. Now in this case, two types of linkages are present in glycogen. One is alpha 1,4 glycosidic linkage and another is alpha 1,6 glycosidic linkage. When two glucose molecules are joined in a straight chain, then they possess alpha 1,4 glycosidic linkage. Now when a branching point is created, as shown in this figure, at that point only the alpha 1,6 glycosidic linkage is present. Now that branching point is further extended by adding glucose molecules by alpha 1,4 glycosidic linkage. So only at the branching point you will find alpha 1,6 glycosidic linkage. So this is about the linkages present in the glycogen, alpha 1,4 and alpha 1,6. Each branch on the glycogen molecule is situated 3 to 4 glucose molecules away from the initial branch, and each branch has 6 to 8 glucose units, or its length is about 6 to 8 glucose units. Coming to the importance of glycogen, it supplies glucose in the post-absorptive phase of the body. What is meant by post-absorptive phase? As far as metabolism is concerned, our body has three phases: absorptive phase, post-absorptive phase and fasting phase. Absorptive phase means it is a phase where absorption is taking place from the gastrointestinal tract, that is after meal. So this phase lasts for 2 hours. During this absorptive phase, large amount of glucose enters in our blood, which is utilized for energy, and the remaining glucose is converted into glycogen and stored in liver and muscles. In the post-absorptive phase, it starts from 2 hours to 12 hours. It may vary from person to person, this phase. The glycogen which is formed or stored in the liver undergoes breakdown. The process is known as glycogenolysis in liver, and supply glucose to the blood for utilization by the peripheral tissues. So in the post-absorptive phase, the glycogen is a source of glucose for our body. So this is about the glycogen. Next is glycosaminoglycans. They are heteropolysaccharides. Two examples mainly, agar and glycosaminoglycans, are there in heteropolysaccharides. Agar is composed of glucose and galactose, and importance is it is used in electrophoresis technique. You will study this electrophoresis during your practical. Now coming to the glycosaminoglycans, it is also called as mucopolysaccharide. The name glycosaminoglycans is due to presence of amino sugars, and the name mucopolysaccharide is given because first time these glycosaminoglycans are isolated from the body secretions, that is mucin, and hence the name mucopolysaccharides are given. Now these glycosaminoglycans composed of two monosaccharide derivatives. One is amino sugar and another is uronic acids in an alternative sequence. Now we will see what is amino sugar and uronic acids. So this is the diagrammatic representation of various amino sugars. First is glucosamine. In this case, at second position, the OH group of glucose is replaced by amino group to form glucosamine. In the next, the acetyl group, that is COCH3 group, attaches to the amino group by replacing its hydrogen atom. So this amino sugar is named as N-acetyl glucosamine. Now in the third figure, sulfate group or SO3H group is attached to the sixth carbon of the glucose along with amino group and acetyl group at second position. Therefore, the name of this sugar is N-acetyl glucosamine 6-sulfate. And the last is, here sulfate group is present at sixth position, as well as it is attached to the amino group instead of acetyl group in the previous cases. Now such sugar is called as sulfated glucosamine. So these are the amino sugars found in glycosaminoglycans. They are derivatives of glucose monosaccharide. Now these are the amino sugars obtained from galactose. Again same, galactosamine, where at second position amino group is attached by replacing the hydroxyl group of galactose. In the second figure, again acetyl group is attached to the amino group, so it is called as N-acetyl galactosamine. And in the third figure, sulfate group is attached to the sixth carbon plus amino group and acetyl group at second carbon. Such galactose is called as N-acetyl galactosamine 6-sulfate. So these are the various amino sugars found in glycosaminoglycans. So this is the explanation. When amino group and acetyl group is attached to the glucose, it is called as N-acetyl glucosamine, and when these two groups are attached to the galactose, it is called as galactosamine. Along with this, sometimes sulfate group is also attached to the sixth carbon or fourth carbon of glucosamine, then they are called as N-acetyl glucosamine 4-sulfate or 6-sulfate. Similarly, when the sulfate group is attached to the fourth carbon or sixth carbon of N-acetyl galactosamine, then they are called as N-acetyl galactosamine 4-sulfate or 6-sulfate. Coming to the uronic acid, this is the second monosaccharide derivative found in the glycosaminoglycan. Here the last carbon atom of the monosaccharide is oxidized to carboxyl group, then they are called as uronic acid. I will show you the figure. So there are two types of glucuronic acid found in our glycosaminoglycans. One is D-glucuronic acid and another is L-iduronic acid. This is the structure of D-glucuronic acid. Just now I have explained. The last carbon of the glucose is converted into carboxyl group. Here you are seeing L-iduronic acid. So see D-glucose and L-glucose, which are mirror images of each other on the left side. Now L-idose is an isomer of L-glucose. You can see the difference at second carbon atom. Now when this L-idose is oxidized at its last carbon, that is at sixth carbon, where CH2OH group is converted into carboxyl group, then it is called as L-iduronic acid. So two types of uronic acids are present, D-glucuronic acid and L-iduronic acids. Now we will see about mucopolysaccharide. Now depending upon the composition of monosaccharide derivatives present in the mucopolysaccharide, there are 6 types of mucopolysaccharides or 6 types of glycosaminoglycans. They are hyaluronic acid containing 4-sulfate or 6-sulfate, dermatan sulfate, keratan sulfate, heparin and heparan sulfate. So we will study one by one. First is hyaluronic acid. It is the largest mucopolysaccharide. It is composed of repeating unit of N-acetyl glucosamine 6-sulfate and D-glucuronic acid. So this is the diagrammatic representation of hyaluronic acid. See, N-acetyl glucosamine 6-sulfate attached to D-glucuronic acid. Again this unit is repeating. D-glucuronic acid is attached to N-acetyl glucosamine 6-sulfate, which is further attached to D-glucuronic acid. So amino sugar and uronic acid is present in alternate manner. There are two types of linkage. One is beta-1,4 glycosidic linkage and another is beta-1,3 glycosidic linkage. Importance of hyaluronic acid. It is present in connective tissues, tendons. Tendon is a part of muscle where it binds with the bone. Hyaluronic acid is also present in synovial fluids, which is present around the joints and lubricate the joints. Also hyaluronic acid is present in the vitreous humor. Vitreous humor is found in the posterior chamber of our eye. Now this hyaluronic acid acts as a shock absorber. The enzyme hyaluronidase hydrolyzes the hyaluronic acid. So this hyaluronic acid forms a ground substance in our body. The enzyme hyaluronidase is present in the bacteria. Therefore, these bacteria easily dissolve the hyaluronic acid present in the various tissues and they can enter inside the tissue by dissolving the hyaluronic acid with the help of enzyme hyaluronidase and leading to infections of the tissues. Next glycosaminoglycan is containing 4-sulfate or 6-sulfate. It is most abundant mucopolysaccharide in the body. It is composed of N-acetyl galactosamine 4-sulfate and D-glucuronic acid. It is also bound by beta-1,4 and beta-1,3 glycosidic linkage. Amino sugar and glucuronic acid is present in alternate sequence. Again, this is the diagrammatic representation where N-acetyl galactosamine 6-sulfate binds with D-glucuronic acid. The same unit again repeats. L-galactosamine 6-sulfate binds with the D-glucuronic acid. So two types of linkages, beta-1,4 and beta-1,3 glycosidic linkage is seen. Now this containing 4-sulfate or 6-sulfate also binds with the proteins. It binds with the core protein with the help of O-glycosidic linkage. In the formation of O-glycosidic linkage with protein, the hydroxyl group of serine mainly or sometimes threonine plays an important role. So by this O-glycosidic linkage, this chondroitin sulfate and protein binds to form a glycoproteins. Now other importance of glycosaminoglycans chondroitin 4 or 6 sulfate is it is present in again connective tissue, tendons, ligaments. Ligament are the tissue which joins the two bones at joint. Chondroitin sulfate is also found in bones and cartilages. Next glycosaminoglycans is keratin sulfate. It is the only mucopolysaccharide which does not contain uronic acid. Now what I have told you, glycosaminoglycans are made up of amino sugar and uronic acid, and they are present in an alternate manner. But in keratin sulfate you will not find uronic acid. So keratin sulfate is composed of N-acetyl glucosamine 6 sulfate and D-galactose in an alternate manner. Again, the two types of linkages are beta 1,4 and beta 1,3 glycosidic linkage. So this is the diagrammatic representation of keratin sulfate. N-acetyl glucosamine 6 sulfate binds with galactose. This galactose in turn binds with next N-acetyl glucosamine 6 sulfate, which binds with next D-galactose. So in this way alternate amino sugar and uronic acids are present. Now this keratin sulfate also binds with the core protein, but the linkage here is N-glycosidic linkage. Now this N-glycosidic linkage is formed between the keratin sulfate and amide group of glutamine and asparagine. This keratin sulfate is also found in cornea and it maintains the transparency of our cornea. Next is dermatan sulfate. As it was first found in skin, hence the name. It is composed of N-acetyl galactosamine 4 sulfate and L-iduronic acid. What is iduronic acid? I have shown in the figure previously. They are bind together by again the two types of linkage, beta 1,4 and beta 1,3 glycosidic linkage in an alternate sequence. N-acetyl galactosamine 4 sulfate binds with L-iduronic acid in dermatan sulfate in an alternate manner they are bind by beta 1,4 and beta 1,3 glycosidic linkages. This dermatan sulfate forms O-glycosidic linkage with core proteins. Again here serine and threonine plays an important role in the formation of this O-glycosidic linkage because they contain hydroxyl group OH. Dermatan sulfate is present in skin, blood vessels and heart walls. It forms various structures of the body. Next is heparin. Heparin acts as a anticoagulant in our body which prevent clotting of blood in the blood vessels. It is composed of sulfated glucosamine and L-iduronic acid. Sulfated glucosamine I have shown in the previous figure how that structure looks like. Now here the linkage is only one linkage, that is alpha 1,4 glycosidic linkage. Heparin also binds with the core protein by O-glycosidic linkage. The importance of heparin is it is synthesized by mast cells of liver, lungs and skin. And then when it enters into the blood, it prevents blood clotting. This is the diagrammatic representation of heparin, sulfated glucosamine and L-iduronic acid in a repeated sequence. Heparin sulfate, it is similar to heparin. It is composed of N-acetyl glucosamine 6 sulfate and L-iduronic acid. They are also bind by alpha 1,4 glycosidic linkage. This heparin sulfate also binds with core protein with the help of O-glycosidic linkage. Majority of the polysaccharides in our body, they are always binds with protein to form a glycoprotein, and the linkage is either O-glycosidic linkage or N-glycosidic linkage between proteins and polysaccharides. The importance of this heparin sulfate is it is found in the basement membrane and also it is found on the cell surface. This is the diagrammatic representation of N-acetyl glucosamine 6 sulfate and L-iduronic acid. They are repeating many times to form heparin sulfate. So we have studied in today's lecture about storage polysaccharide, that is glycogen, and structural polysaccharide, that is glycosaminoglycans. In these glycosaminoglycans we have studied 6 glycosaminoglycans which are given in the syllabus. Thank you very much.